My name is Shad Samir, and I will start talking about the antibacterial and biodegradable tissue nano-adhesives for rapid wound closure and healing. For the wound healing and the closure, this process involves the replacement of damaged or destroyed tissues with the newly produced tissues. This process or the reconnection of the tissues is a very hard process, and it plays a real challenge due to the complexity of healing itself. Here, various tissue adhesives are used for the wound closure and for the wound healing, as it facilitates the wound healing, and it also protects and put limitations to the toxicity and the infections. It's highly desired to have an ideal tissue adhesives which employs biocompatible materials for educate and rapid addition, accelerated wound closure and protection against infections, in addition to improving the mechanical properties and adhesive strength. The nanoparticles that are used as a tissue adhesives are called the inorganic nanoparticles. They use the nanobridging effect between the protein chains in the wound, as this will help in the wound closure and healing. They include the silica nanoparticles, magnetic nanoparticles, and the metal nanoparticles. Those nanoparticles exhibit a strong adhesive properties comparing to the traditional tissue adhesives. For the type of nanoparticles, first we will start with the multifunctional nanoparticles, as this type will integrate both the nanobridging effect and the antibacterial effect, and this will guarantee a safe and an efficient wound healing. The second type is called the mesoporous silica nanoparticles, MSNs. They received a considerable attention due to their highly specific surface area, as this will result in increasing the functionalization capabilities and compatibilities. The third and the last type of nanoparticles is called the silver nanoparticles. They consider to be an efficient and an excellent antibacterial tissue additive. Here we will be dealing with the problem that is related to the slow silver ions releasing. This will affect the practical application of the AG nanoparticles, and to solve this problem, the AG nanoparticles are embedded within the MSNs, and then AG MSNs will be synthesized and used as a tissue adhesives. The AG MSNs are used to protect the AG nanoparticles from aggregation and to also control the releasing of AG ions. This will add a disulfide bond to the MSNs and will facilitate their clinical translation. The AGMSNs also are an effective and a safe tissue adhesives with an antibacterial and degradable properties. This will help in the wound healing and the closure. The AGMSNs are synthesized by the controlled reduction of ultra small nanosilvers. This will happen on both the surface and the large pore of biodegradable MSNs. For the characterization techniques that are used, the transmission electron microscopy, the energy dispersive X-ray spectroscopy, the X-ray diffractometer that is used for X-ray powder diffraction investigation, the BET method, which is used for calculating the surface areas, the total pore volume and the pore diameter, and also the zeta sizer for the size distribution. The adhesion strength test is done to compare between the MSN and the AGMSNs. This test is done by using the gelatin hydrogels that was selected as a traditional model to test the addition to a biological tissue. And the biological tissue that is used here is a cow liver. For the preparations of this test, this will include dissolving the gelatin into a deionized water and spreading it into a glass plate for over a night. Cutting the gelatin films into a specific size and attaching the two films. Then pressing for 30 seconds after adding the two types, which is the MSN and the HMSN solutions. The tensile test is done and the failure forces for each type is calculated. Experiment in this paper will be done by using rats. We will have 60 groups, and each group will have their left one sutured and the right one is closed by an MSNs and AGMSNs as a tissue adhesives. Microscopic images are taken for the one side for 1, 3, and 5 days. Blood samples are taken and a hematocytometer is used for the hematological parameters. The hematocytometer is an instrument that is used to count the cells in the blood samples or the fluid that is studied under the microscope. All the images are studied and examined under a digital microscope. The results of this paper will be summarized as the AGMSNs will protect against the aggregation of aging nanoparticles, and this will be protecting also from the bacterial growth and the toxicity. MSNs exhibited similar physiochemical properties to those of the AGMSNs. The failure force and the addition force for the AGMSN in both ways is higher than the MSNs. For the addition force for the both type, it will be concentration dependent as it will be increasing with increasing the nano-adhesive concentration. Completing for the results, the HMSNs and the MSNs are both suitable as a nano-adhesives and as tissue adhesives for wound closure and wound healing. Both exhibits excellent antibacterial activity against the added bacteria. Both showed an overall faster and a time-dependent degradation, and also the AGMSNs achieved an easy wound closure without any infections.